away from studying genomics, I mean the core genomic sequence, we have to realize that every aspect outside that has a temporal component. In a sense, our physiology is constantly humming. So much of my work focuses on bringing the time dimension into environmental health studies. And today I'll describe some technologies that we are developing, but I wanted to do it in the context of the theme of this symposium on aging, so I will use ALS, or Lou Gehrig's disease, as a case study on how to overcome some long-standing challenges in studying that hum or that temporality in environmental health. Before I do that, I just want to declare I have some conflicts of interest. I'm not embarrassed at all. I'm actually quite proud of them. My company is a co-sponsor here, and I'm very grateful for the opportunity to be able to support this symposium and others like this in some small capacity. So a little bit about amyotrophic lateral sclerosis. It's not a common disease. It's actually a rare disease, but it's a terminal disease. And it affects upper and lower motor neurons, so you start losing various muscular functions, and people usually die from respiratory failure. There are about 5,000 new cases in the US and 2,000 in Japan, and the median age of survival from diagnosis hasn't improved at all. Once you find out, it's about 5 years. The sad reality is that finding out itself is a journey that takes on average about 1 to 1.5 years. You spend, at least in the US system, tens of thousands of dollars out of your own pocket to get this news, because there are no real biomarkers for this condition. Since both Japan and the US have a love for baseball, one of America's famous patients who got this disease was Lou Gehrig's, and as fate would have it, I actually lived not too far from where he used to live in Larchmont, which is a suburb in New York. So it's also called Lou Gehrig's disease. This is a brutal condition, and we struggle in every aspect of it. We struggle to detect it. There is no such thing as a screening tool. We also struggle to find treatments. A few years ago, there was just this public outcry, because it's such a brutal disease, that there was some glimmer of hope behind a company called Amylyx. It's a real wonderful story of about 2 young academics who thought maybe there's something here, and they started a company. So the FDA, under public pressure, gave an approval to a drug, and there are very few drugs for ALS. 2 years later, this drug was withdrawn from the market. I'll tell you why, for me, this is a real point of contention, and why there is a solution to this problem, which I'll discuss in a moment, but the solution lies in the time dimension. One of my favorite papers to discuss was from our colleague Steve Rappaport, who in many ways helped popularize the concept of the exposome. I would argue that if it wasn't for his paper in Science, we probably would be calling it a different name. He did a large study in Europe comparing heritability. And sure, heritability and genetic risk are not the same thing, but it's still a good way to convey a real disparity in how national programs have approached different disorders. So all the dark bars represent the non-genetic component, and the light gray bars represent the genetic component. So if you look for ALS, only about 5 to 10% of it is explained through heritability. The rest is explained through non-genetic factors. I actually don't like this dichotomy. I like the way Dr. Wright described it once to me, and I think he knows who originally said it, that there is no such thing as a genetic disease or an environmental disease. Every disease is a genetic and an environmental disease. But my argument here is most of the focus on ALS has been on genomics. Most of the funding on ALS has been on genomics until recently, where we are now more and more interested in the environmental determinants of this condition. I worked for a while with this very prominent group out of the University of Michigan, and they, Claudia, who is now actually at the NIH, she was a postdoc there, and then she left to join the NIH. developed this model. And she said that early in life there are genetic underpinnings, sure. And there's all of this exposures and what she called dyshomeostasis or dysregulation. And then on top of this, there is epigenetic factors and biological response. For example, inflammation. And the onset is much later in life, after 50s and 60s. The average age of diagnosis in the US is around 60 years of age. So herein lies a real problem. Like I mentioned, it's the time dimension. If you're going to detect the disease in their 60s and we're saying the environmental hits happened in early life, how do I go back in time to figure out what your exposure was? I'm looking at Joel Schwartz and thinking, what was your exposure back when you were my age, which is 26? Here's another problem. It's not just that there was an exposure, it's also what's inside our body. The biological response, the way we react to things is very different than if you're on the left side of this and living a very good life as a fetus. I always say, the moment you're born, it's all downhill from there. Right to the right-hand side of this graph where, well, things are less certain for your future. So how do we go back all that time, decades, and figure things out? And again, I'm saying the same point, but really you want to do a case control study for a condition like ALS. So here's a quick look of one idea that I've been working on for almost 20 years, and many of my US colleagues have seen this slide before. The idea is a very simple one. If you've ever listened to a vinyl record or looked at a tree that's been cut down, or even thought about how hard drives work in a computer, the idea is actually exceedingly simple. If you can find an incremental archive, then you have an entire history there, because that's how incremental archives work. If you map behind the growth rings in a tree, you're just moving through this incremental archive and going back in time. And in my previous career, I used to be a dentist, and one thing that was very useful from that career, about the only thing that was useful for me from that career, was realizing that teeth have growth rings in them. So there are growth rings in teeth forming every day. So now that you're here eating sushi and seafood, there are components of seafood being deposited in your teeth, which when you go back to New York and go back to the American diet, you will see that switch. I give this example from Bob's cohort in Mexico. We collected this tooth as 10 years of age, but you see how if you look at the x-axis, we've built a weekly timeline of exposure going back into even prenatal life. Because here's the other amazing thing about baby teeth. They start developing in the second trimester. So every time a child sheds a baby tooth, they're actually handing you fetal tissue. And I collaborate with Shoji-san in the JEX study. We are going to analyze about 40,000 teeth. So you can imagine all the wonderful data one day that will come out from. And Miyuki-san is sitting there who's leading the analysis. But we don't just measure metals anymore. We measure lots of different things, compounds outside your body, the structural building blocks that are in the green square, and also our biological response, inflammation, oxidative stress. And this allows us to do a whole new type of molecular phenotyping beyond what genomics alone can do. Because in the same tissue, you're measuring the exposure, what your physiology is like, what is the biological response to that, but you can add a time dimension. I saw Dr. Wojciech present this sort of an equation of precision environmental medicine, which had genomics, epigenomics, and omics. I'd like to add the time dimension to it. I won't go into a lot of detail, but I spent many, many years. I was lucky enough to get an R35 grant from the NIEHS, which allows me to do that, to study just the basic concept, the physical concept of time as it's applied to human health. I would argue that most of the work being done in physics hasn't even scratched the tip of the complexity, the true complexity of time, because the only real complex systems in the world are not those ones that we're studying in planetary motion, but actually it's our biology that is the only true complex system. I've written a bunch of concept pieces, even a book. Those who win the poster prize, that is your prize. I apologize in advance. But this is the year of the cicada, so if you don't want to read it, you can always sort some papers. I call that approach an environmental biodynamics approach. I won't go into too much detail about that. Like I said, I want to convey the importance of the time dimension through a case study of ALS. I'm going to present two technologies. The first one is just using teeth. This is a very small study. You will see 36 ALS cases and 31 controls. Many studies in ALS are small because it is a rare condition. It's also not perfectly balanced. There are some differences in ethnicity. The way the condition is, it impacts white men much more than other ethnicities. Also, our controls are a bit younger. But moving beyond that, this is just a demonstration of, hey, we're recruiting patients of an average age around 60. How are we going to determine their early life environmental risk factors? So we use that idea. We got their wisdom teeth. We got whatever teeth that they had during their dental treatment, because one thing that happens during ALS is you can't really look after your dental health very well. You start losing teeth. In fact, dentists will extract even a few healthy teeth if that's all you have left, so that you can get a denture. So somebody else can clean that denture for you because you no longer have the ability. So you're getting teeth as part of their dental treatment. But whenever I look at this graph, it still excites me to see how useful this basic biology of a tissue that is so easily discarded, just your common tooth, allows us to achieve this. If you look at the x-axis, you will see that we've gone back to early life, the first 15 years of life, and built at a weekly scale different components of what we call a metal mixture. I'll show you each mixture. I owe a debt of gratitude to Dr. Chris Jennings and Dr. Galip Bello, who were Chris Jennings is still at Mount Sinai, Galip used to be, who actually developed a statistical tool called lagged WQS, or weighted quantile sum regression, specifically for the tooth data. What you see here is that even at birth, there is something about your physiology that's absorbing a lot more metals. This is how I like to tell the story, but that's all it is. It's such a small sample size. By the age of 5, something is trying to bring that back under control, and you're kind of under control up till around 10, 12 years of age, where things tend to start going haywire again. And we see that if you look at individual metal exposures, early on there's a high uptake. Things tend to settle down, but afterwards it's all going all over the place. So there is systemic elemental dysregulation, as I like to call it, as an early feature of something that's going to show a clinical sign, even an early sign, like foot drop or a tremor, decades later. We did an animal model as well. There are not that many good animal models, but if you knock out the SOD one and introduce a mutation there, you can start having mice that kind of behave like ALS mice, and we see those same metal mixtures having an impact. So we went quite deep into understanding the pathology of ALS. By now you'll realize that there's a critical flaw if you want to move this into a clinical setting. You can't just have a patient sitting in front of you without any severe symptoms and say, hey, hand me a tooth, right? That's just not going to happen. So over the past few years, I've been thinking of what other tissue in the body is an incremental archive that is easy to get. That's something that you can get straight away but carries the time input. I won't go into the detail, but more and more I work with some hardcore AI scientists. We realize if you want to really characterize the signal of certain diseases, especially disorders of the brain that have no biomarker, and the reason they don't have a biomarker is you need 500 to 1,000 time points. This is not the place for a deep AI discussion. I'm not the right person for it, so I'll just convey it in this very simple sense. Imagine if you have a song, but I only give you one note. Can you identify that song? Or if you were doing a prospective cohort study on that song, and it's a 3-minute song, and every 30 seconds I just let you listen to a note, would you be able to figure out what that song is? And the answer is no. So what we needed was a sample that's easy to collect, that's stable, so we can do studies in poor regions where you might not be able to collect blood or anything, but have about as much information as 500 sequential blood samples. The answer is that you can get all of that from a single hair. Now, the irony that I'm talking about hair biomarkers is not lost on me. Joel, you're not that far behind, so you don't laugh and Dr. Wojcik doesn't get to laugh. All can. Turns out hair are actually very hard to analyze because they come in all sorts of different shapes and sizes. No two hair are alike. I worked with Shoji-san, the JEC study on Japanese baby hair. It's so fine that if you're holding it, sometimes you're not even sure you're holding one, as opposed to some very thick and curly hair. And I want to deliver this at scale. Luckily, I work in the private sector as well, so we raised some money. We raised a lot of money, I should say, actually, because it would have taken me about 15 R01 grants to get the same amount of money. We built a high-throughput robotic platform. This platform does it. Not only does it pick up hair and get it nice and straight without breaking it, it actually opens up hair. Because the information, the biological signature is on the inside. If you imagine layers, it's in the core of the hair, without raising temperature. But if you heat up hair, you're losing all those molecular signatures. And I work with a physicist who's developed a whole new class of lasers to analyze that hair. We generate huge amounts of data. We can measure thousands of chemical signatures, but at 500 to 1,000 time points. For each individual, you have 500,000 to 1,000,000 signatures coming out from their hair. The person never even has to come to a clinic. They just cut their hair at home. We have a collection kit. We give them a very detailed instruction how to cut that hair, put it in a box, and just ship it to us. And we use machine learning and some AI tools to just look at that hum, as I like to describe it, those patterns that are very characteristic of certain conditions. This is a much larger study. I owe a debt of gratitude to the folks at the CDC, but if you look at cohort 2, it's all cases, but this is a national collection. They collected hair from cases around the country. And then we have two centers here, one other center here, which is at Dartmouth University, where they have cases and controls. So around 300 patients for an ALS study is now getting into the realm of a better size study, given that it's a rare condition. This is what we start seeing in healthy subjects. We actually have this complex structure that, again, using machine learning, but these bars that are connecting different elements are not just concentration. In fact, concentration doesn't even come into the picture. This is all studying the dynamics. So how are you connected over time rather than how are you connected over concentration? I want you to look at what happens in an ALS patient. The complexity is actually broken down. We see this drop in complexity in other neurological disorders as well. We saw this in autism and ADHD. It's just different components of these molecular interactions are involved. As an example, if you look at manganese in a healthy patient, it has this centrality. It's sort of a conductor in this symphony, whereas in an ALS patient, it's pushed to the side. And we know from the SOD1 mouse, SOD1 is an enzyme that is manganese dependent, that manganese metabolism is disrupted in ALS. So we're seeing something very useful. We want to convert this into a clinical tool, something that helps clinicians diagnose ALS. Think of it like a cholesterol test for heart disease, something that helps, but by itself it's not going to be accurate enough ever. But it's a complement. These are very early results. I just want to be a good scientist and say, from a small cohort study with an imbalance in cases and controls, this is probably overly optimistic. And whenever I look at this ROC curve, I think the reality will probably be not such a good performance. But for what it's worth, this is the data we have, and it's showing us pretty decent clinical performance if you look at negative and positive predictive values. You can also start looking at severity. Now we are seeing some very good separations in those who have a high severity version of ALS and low, and there are different ways to measure severity. One of this is, it's a score called the ALS severity score, and we are explaining about 45% of the variability in this. Again, it's early days, we need to replicate this in other studies. We are doing a large study actually in Sweden. To a center at the Karolinska, we have a nationwide study where we have about 500 patients, and we have collected hair from their siblings and someone living in their neighborhood, so genetically related person and unrelated person. So we have about 1,500 samples to go through, which we are working. What I really want to summarize here is not so much the ALS story itself, but to convey that one aspect of our health, brain health, we are lagging way behind. The answer does not lie in a genomic-centric approach. Whether you look at autism or ADHD or schizophrenia, how do you know you have that condition? Because you start behaving like that. Right? Someone looks at your behavior and says, oh, you behave like an autistic person, or you behave like a schizophrenic, or you're behaving like a person with ALS. That's why progress has been so slow. For me, the answer really lies in this realm of environmental medicine. And a question that isn't being asked often enough is, what is the molecular definition of ALS? In fact, what is the molecular definition of autism? 3% of our population now suffers, has autism or are autistic. Yet if you look at the CDC definition, it's all, well, you have repetitive behaviors, you have trouble with social interaction, things like that. Would we accept that for diabetes? Would we go to our doctor and the doctor said, yeah, you behave like a diabetic, so I'm going to give you this prescription or I'm going to call you a diabetic? No. So what is the molecular definition of ALS? And how do these clinical trials plan to succeed? The Amylyx trial failed not because we know for sure it works or not, there is no molecular endpoint. So how do we know this drug is working or not when we're not measuring disease in a true molecular sense? And again, by molecular, I don't mean genomics or just proteomics. I actually mean much more exposomics, because that has been completely missing. All these other approaches have been tried, and they did not succeed. Right? I felt so strongly after the Amylyx disaster or failure, that I actually wrote an opinion piece, but not for academia. I purposely wrote it for the pharma industry, said, hey, you guys are missing something. We in academia are already working on. It's time to start using these tools. Instead of collecting a blood sample at the beginning of a trial and the end of a trial, just collect hair, and I have 2-hourly resolution. So you have no missing gaps in a clinical trial. As long as you collect hair at a few time points, or you have one hair that is long enough, I can pretty much give you multiple measures a day, a molecular movie of sorts, rather than just a snapshot. So going back to something that multiple earlier speakers have said, that we must work towards a global map of the exposome, just like we did a global sequence or a global genome project. To that, I would just like to add that if you sequence the exposome globally, if you don't include the time component, it really will be an incomplete map. So we really want to have a global temporal exposome. I'll stop there. I do want to acknowledge the NIEHS and the CDC for all the grants. Really the kind of work I do, high risk, potentially high reward, would not be possible if the NIH and NIEHS did not have schemes like the River Grant that allow us to take these risks. Thank you again to the organizing committee for this opportunity.